So I'm talking about programming in living cells. Not recently, but in the past couple of years, we discovered something called CRISPR and Cas9. These are found in the immune systems of bacteria. They were found to be a magical tool that allows for you to change the genetic sequence of a DNA strand very remarkably easily. We've been using this technology to great success already. For example, one of the biggest things we've done with it is the COVID-19 vaccine, where they took the antibodies for COVID and made a CRISPR molecule that they injected into us that allowed for to reprogram your white blood cells to be able to recognize COVID and effectively allow you to become immune to it much more quickly than if you're to fight it without a vaccine. The way it works is kind of like it's kind of like a little, it's pretty straightforward. You can cut up the DNA and replace things. You can remove things, you can and replace them. It's kind of like a text editor. It's a little bit more complicated than that, but it's a little bit too in depth for a quick chat. So we currently are trying to find better ways to use it. And the problems we have with it at the moment is that it's kind of like writing in bytecode, because you're kind of just dealing with the raw DNA sequences. You don't actually, it's very hard to tell what it actually does. Most of the programming we've done is pretty much guessing what chunk of DNA does what, and then just shoving that in there and hoping for the best, and through experimentation, getting it right. It also requires a lot of understanding of biochemistry, because you need to understand what chemical reactions occur once you've changed the DNA. The way DNA works is it will, in your cells, it gets read and it creates proteins, which then will do all the functions of your cells. And so you have to understand how all those processes work to get your functions working. And it requires a lot of manual lab work. You have to spend hours and hours just setting up files with different cells and making sure, just continual testing, lots of different iterations. So I was looking at solutions to this, and I found one, which is a high-level programming language for DNA called Cello. It's almost like an assembly language for DNA, kind of makes it a bit more human readable. And then once you've got it in a human readable format, it will kind of compile it into a DNA sequence. And because it's human readable, it requires a lot less knowledge on the inner workings of a cell. The only problem with it is it isn't a full-blown solution. It creates logic circuits, kind of like how a computer processor would work, rather than how living organisms work. They have a set of different proteins that it's pre-programmed to use, and it combines those to create your tools. It's really powerful, even with that limitation, because you can create quite complex biomachines with it. You still need a lab to do it, because you get the DNA sequence, but you still have to fabricate that DNA and put it into cells. And when I was doing research, I found that it only works on E. coli bacteria, and they haven't found a way to get it to work on other cells yet. In terms of the future for this, it's an amazing tool. It's very powerful, and we kind of just stumbled across it. It's something we don't know the limits of at all. We've pretty much got a tool that we've kind of got like, what's a good analogy? Got a computer for a caveman. We have no idea how to, we kind of figured out how to use it, but we don't know how, we don't have the knowledge to use it to its full extent yet. And I'm excited to see how scientists manage to get it in the future, manage to use it in the future. That is my talk.